Welcome everyone. Today we are going to learn about action potential. This is a graphical representation of action potential. The x-axis demarcates time in millisecond and the y-axis denotes membrane potential in millivolt. Now what is membrane potential? The difference in voltage inside and outside of a cell is called membrane potential. Suppose this is the plasma membrane, so we know that plasma membrane surrounds a cell and it creates a boundary. The outer portion of the cell is called exterior or external environment and the inside is called cytosol. Now, if you insert a microelectrode inside a cytosol and if you keep another microelectrode outside and if you connect these two microelectrodes with a potentiometer or voltmeter, you will get a reading. In normal condition, that is in resting condition or unexcited condition of a neuron, you will get a value. This is called membrane potential, or also we call it electric potential or voltage gradient. Now voltage gradient means the difference in voltage. So the y-axis here denotes the membrane potential change in millivolt. Now as you can see, I have drawn this graph in four different colors. One is blue, red, green and a small portion in black. And I have written the four different parts of this action potential in these colors. Like the blue line represent resting membrane potential. Now what is resting membrane potential? When a neuron is in unexcited conditions, this is called resting membrane potential. This is the baseline of the membrane potential or the voltage gradient present in an unexcited neuron. Basically, this resting membrane potential actually not part of the action potential, but it is the baseline. Now, when the membrane potential increases above the resting membrane potential, now in case of neurons, the resting membrane potential can vary in between minus 40 millivolt to minus 90 millivolt. Suppose this resting membrane potential is minus 70 millivolt. Now when the membrane potential increases above the resting membrane potential, we call this is depolarization. So the red line denotes depolarization. I have written here that depolarization is greater than resting membrane potential. Now these depolarizations actually stop at a certain value, or we call it peak. Here I have mentioned that is plus 40 millivolt, but in any way it can reach up to plus 50 millivolt. So there is a peak and it stops. So depolarization stops. Now when the depolarization is at its peak and it falls up to the resting membrane potential. We call it repolarization. The green color represent the repolarization. So there is resting membrane potential, depolarizations, and after reaching the peak of this membrane potential, it falls. It is called repolarizations, and this represents up to the resting membrane potential. Now for a very brief period of time, less than milliseconds, also 1 millisecond, this brief period of time, the membrane potential actually falls below the resting membrane potential also. See here the resting membrane potential, the black line actually goes down of the resting membrane potential. So this portion is called hyperpolarizations, and this membrane potential is less than resting membrane potential. You have to understand this. This red line, green line and black line represent action potential. But always the membrane potential comes up to the resting membrane potential because this is the baseline of the neuron. Basically all animal cells have resting membrane potential, but neurons also has a resting membrane potential. Now in between subsequent action potential, there is a period called refractory period or recovery period, and that is actually 4 millisecond in time period. Now if you ask me the time point of the action potential, in case of squid neuron, it takes about 5 milliseconds to depolarize, repolarize and hyperpolarize. But in case of mammalian myelinated neuron, it takes just less than 1 millisecond also. But the refractory period is actually greater than the total action potential, the depolarization, repolarization, hyperpolarizations. It takes about 4 milliseconds. Now in this period, you cannot excite a neuron further. So in between subsequent two action potential, there is a gap or there is a period of time in which action potential does not occur. So we call it refractory period. It is about 4 milliseconds. Remember this. Now, in another way, we can also divide these three potential, because in this potential the membrane potential of the resting membrane resting condition actually changes. Okay, so in case of depolarization it rises, in case of repolarization it falls, and hyperpolarization means less than resting membrane potential. Now we can also divide this action potential in different phases, like when the membrane potential actually increases above the resting membrane potential, this line we call rising phase up to 0 millivolt. But when the membrane potential actually becomes positive in value, so we call it overshoot phase, and here it actually touches the peak point. Now when it reaches the peak point it actually falls, and we call it falling phase. And here the small period where the membrane potential actually goes down of the resting membrane potential, we call it undershoot phase. Now you can relate undershoot phase with hyperpolarization. So I have drawn this in black color. So basically you can divide this action potential in phases also, and this is the main theme, that is it contains three important phases: one, depolarizations, repolarizations, and hyperpolarization. Now one thing I want to mention that this is a typical action potential graph of a neuron, but the action potential graph can vary. Now let's see how action potential graph changes in different conditions or situations. Like if we measure action potential in myelinated actions, now this is a typical structure of a neuron. This structure is called axons. This is the cell body, and these projections are called dendrites, and this is axon termini. Now, if we measure action potential in the axons of myelinated actions of a vertebrate motor neuron, you will get a graph of action potential, a typical graph where this is the resting membrane potential, this red line indicates depolarizations, green line is repolarizations, and this short period of time is called, this black line is called hyperpolarizations, and ultimately it goes up to the resting membrane potentials. So in case of myelinated actions of vertebrate motor neurons, you will get this kind of a typical graph of action potential. But if we measure the action potential in the cell body of the same motor neuron, same motor neuron, that means here if we measure the action potential, you will get a different kind of action potential graph. As you can see, this graph starts with depolarizations, there is no resting membrane potential, and repolarizations, there is no hyperpolarization and resting membrane potential. Now basically if you see the amplitude of the action potential, you will find a difference. In case of action potential that we have measured in the axons, you will get a positive 40 millivolt membrane potential. This is the peak of the action potential, but here you can see it can cross 0 millivolt or slightly more than 0 millivolt. So it will not touch the plus 40 millivolt. So the same motor neuron, if we measure the action potential in the cell body and in the axons, they differ. Now let's take some examples of other cells like cardiac myocytes, that means heart muscles. In the heart muscle there also action potential occur, but if you see the graph you will get a different type of phases. Like in case of myelinated neurons, I have mentioned that it only takes less than 1 millisecond. Basically it takes less than 1 millisecond, but in case of cardiac myocyte the action potential is longer. It can take 150 milliseconds to 200 milliseconds also. Now this longer action potential is important because that longer action potential actually helps in the cardiac myocyte contraction. Now if we divide this whole action potential, we will find 4 different phases. Now the phase 0, this red line is actually rapid depolarizations, okay, like this red line. Now phase 1, this green line actually tells the repolarizations, but in the repolarizations there is different phases. Like Phase 1, this is the notch, depolarization notch. That means here the peak actually falls. Here the peak goes up to that point. That means here in case of cardiac myocytes, it can go up to plus 60 millivolt and then it falls. This is called depolarization notch. In this phase, when the membrane potential slightly fall, not fall sharply, here the fall is sharp, but here the fall is actually gradually occurs. So the membrane potential falls gradually. We call it plateau phase. It is also repolarization phase, but it not falls sharply. Now in the phase 3, this is the sharp fall. Here we will call it repolarizations. And this is the phase 4, that is resting membrane potential. Here also hyperpolarization occur and it actually recovers into resting membrane potential. So if you see, you will also get the resting membrane potential in case of cardiac myocyte is less than 90 millivolt. Here it is approximately minus 70 millivolt. The peak of the amplitude of this cardiac myocyte is different, and the duration of the action potential is different. But basically, this two graphs also shows depolarization, repolarization, hyperpolarization, resting membrane potentials, but in different phases. So that differs. Now, the last example I will show is that if we measure action potential in the dendrite of cerebellar Purkinje neuron. Now I will show the Purkinje neuron how it looks, because it has an extensive dendrites, not like this. It has extensive dendrites. So it can make many contacts. We call it synapse. So when an axon termini of one neuron make contacts with other dendrites of other neuron, we call it synapse. That junction is called synapse. So in the dendrite of cerebral Purkinje neuron, there is many synapses present. So what happens? You will see that complex spikes. Now what is spike? Basically, this action potential is also called spike. Here, this shows a single spike, but in case of cerebral Purkinje cells, you will find complex spikes. There is many action potential exactly simultaneously occurring, because if we measure the action potential in the dendrite of cerebral Purkinje cells, we will get the summation of multiple discrete action potentials generated by different synapses. So in the extensive dendrites, there are multiple synapses coming from different neurons, and that synapses create different kinds of action potential. This is the summation of the action potential. Here also you will find that this is the resting membrane potential. There is a depolarization and repolarization, but multiple times it actually occurs and fluctuates. Ultimately, go to the resting membrane potential. So what I want to mention here, that don't think that this is the ultimate action potential graph. The action potential graph can differ, but the theme is same, that there is a resting membrane potential, the baseline. There is a depolarization, that means when the membrane potential increases than the resting membrane potential, and there is a fall, that is the repolarizations. There is a hyperpolarizations can occur, that means the membrane potential can go down the resting membrane potential and then it actually recovers into the resting membrane potential. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching.